Hi, everybody. In this video, we are going to be talking about a type of tissue as well as a type of organ and organ system. This content comes from multiple places within our textbook. And so I want to start off with a little bit of a lecture outline. First, we are going to make the jump from what we've been talking about, which are cells, right? Cells doing protein synthesis, cells generating energy. So cells essentially functioning for cells' own purposes, to cells functioning as part of a greater whole. So an individual versus part of a community. And so I'm going to use an example of cellular response to tissue damage. So if cells over here are damaged, how do the healthy cells over here respond and modify what they're doing in order to kind of fix the tissue damage? So this will require some communication between cells. It'll require some very intense activity of the cells themselves. And so once we have a basis for understanding how the cells can actually work together as tissues, right, instead of one cell for one cell's sake, one cell for the community's sake, I'm going to show you and introduce you to the integumentary system, the skin system. In this particular lesson, we're going to focus on the epidermis, so the very superficial part of the skin, what we see when we look at each other. We're going to see how the epidermis is made of epithelial tissue. It's broken down into a couple different structures, a couple different layers. And so we're just going to walk through the epidermis as a whole. And then we're going to kind of zoom out. We're going to see what epithelial tissues are in general and where they're found throughout the body. Okay, so just to remind you, in the levels of organization, which we talked about in the beginning of the semester, cells work together as tissues. Okay, so far in this class, we've talked mostly about how atoms come together to form molecules, molecules come together to form cells, and now we see how cells come together to form tissues. Here, if we take a section through the skin, the very surface of the skin, we can see that there are many cells here. Each of these little blue dots is a blue nucleus. So many of these cells come together to form a tissue. This tissue happens to be stratified squamous epithelial tissue. We'll get into that. And this forms the layer of the epidermis. All right, so here is epithelial tissue, and deep to this epithelial tissue, we see that there are two other types of tissue. One is kind of smooth, the other one is pretty patchy, and densely packed proteins that are patchily put together. And so this structure as a whole, so epithelial tissue plus two different types of connective tissue, these tissues are working together to form an organ, and that organ happens to be our cutaneous membrane or our skin. And so those are the levels that we're going to be kind of dancing around a little bit today. So let's start to make this leap across the levels of organization. When a tissue is damaged, or in this case, even when an organ is damaged by a paper cut, for example, right? So this used to be epithelial tissue in the epidermis. This was lots of different types of connective tissue in the dermis. And there's a wound, right? And so that is going to damage some cells. It's going to shear some of those cells completely off. And so this damaged tissue or damaged region is surrounded by cells over here and over here that are perfectly healthy, right? They're just doing their thing. But they must first be aware that there's a problem, right? They're aware that the neighbors in their community, the other cells within the tissue are damaged, many of them are missing, and that ultimately leads to a vulnerability, right? So this opening in the barrier of our skin allows an opportunity for bacteria and other things to get into our body, into our blood, and therefore form an infection. And so, first and foremost, cells have to be able to talk to each other in order to know that, hey, there's a problem over here. We need to modify what we're doing to fix the problem. And beyond that, they need to be able to respond to that message, right? Not only do they need to know that their friends are gone, their friends are damaged, they need to be able to in this case, multiply, right? Make copies of the healthy cells to fill in that gap, right? More and more cells need to fill in this gap and ultimately close up that space, and if we're lucky, return that barrier back to its original shape, its original function. Right, of course, we know that after we get a cut, the skin might look a little bit different for a while, and if the cut was bad enough, it might leave a scar for a long time. And so this is merely a product of the tissue just working so very hard to fill in that gap as quickly as possible to close that barrier. And so the cells actually make a lot more collagen than they usually do, leaving behind that lighter color of the skin that we would call a scar. Okay, so let's break down these processes. First and foremost, the cells need to know that there's a problem, right? Their friends are gone, their friends are damaged, and so cells need to be able to talk back and forth to each other. And so just like humans can communicate in many different ways, of course, we can verbally talk like I'm doing right now. We can use facial expressions to communicate our emotions, our intentions. We can use body language to communicate as well. We can use written text, right? So humans in general have lots of different ways of talking. Our cells are a little bit simpler. Our cells talk with chemicals. So in this image down here, we can see that this cell has something to say, and it releases chemicals. So these chemicals might be molecules. They might be proteins that were transcribed, translated, processed, packaged up by the Golgi, and released out into the extracellular fluid. All right, so they might be that. But these signaling molecules, we call them ligands. Okay, like any kind of signaling molecule is called a ligand. And so these are released out into the extracellular environment, maybe even into the blood. And so the other cells have to be able to hear this message, right? If you're blind, if you're deaf, if you can't read, whatever, like you don't get the same messages from talking, verbalizing, writing, whatever. So cells, the way that they listen is that they express receptors on the surface. So receptors are proteins. We know that proteins have a very particular shape based on their genes. And so this allows receptors to be wildly diverse and be able to bind to specifically and tightly very specific signaling molecules or ligands. Okay. And so here are just some examples of proteins that are considered to be receptors. We see that they're all embedded within the phospholipid bilayer. Here's extracellular space, so outside the cell. Here's the cytoplasm inside the cell. And these proteins, they all have different shapes. And so this one is epidermal growth factor receptor. So that means that it binds specifically to the epidermal growth factor ligands. And so when those epidermal growth factors bind to this very specific receptor, it's going to induce a pre-programmed response within the cell. Of course, all the ligands, all the messages must be a different shape, right? All the words that we have different sounds and therefore have different meanings, convey different meanings. And so this one is an insulin receptor. So insulin has a different shape than epidermal growth factor, and beta adrenergic receptor, this one binds to adrenaline, right? So all different shapes. Which of course is a function of proteins, right? And so again, once the ligand binds to its receptor, right, the message is sent, the message is received by the receptor, kind of like a lock and a key. This induces some kind of pre-programmed response inside the cell. Oftentimes that involves changes in gene expression, right? So we turn on different genes. We say, start transcription, translation, etc., of a particular protein that we weren't previously making, or at least make more of it. Sometimes there are other cellular responses, right? Maybe we will start to break down glycogen to liberate some glucose into the blood. Maybe we will open an ion channel, right? So whatever it is, this particular message binding to this particular receptor is going to induce a pre-programmed response within the cell. And so we have a lot of different types of talking back and forth, of signaling by ligands. And usually this is classified according to kind of how far away the talker and the listener are. First, we have synaptic signaling. This is usually done between neurons and another neuron, or a neuron and a muscle or a gland. So this is signaling right next door over a synapse, which is a couple nanometers wide, so very, very, very close. Synaptic signaling happens lightning fast because as soon as those ligands are released, they diffuse very quickly across this tiny little space and can immediately bind to the receptor. Paracrine signaling is talking back and forth between cells in the same tissue, so nearby but not right next to each other, so within the same tissue. Endocrine signaling, we talked about a little bit before this semester. Endocrine signaling requires the blood. And so here we have one cell needing to talk to another cell all the way on the other side of the body. And so it uses the bloodstream, the blood circulation, in order to carry these messages. Now, the thing about endocrine signaling is that the blood, of course, goes through pretty much every tissue of the body. And so these ligands are going to be encountering all sorts of different tissues. And so how does this message only get to the target cell? Well, the message actually goes past, right? It encounters all sorts of different cells, but those cells aren't necessarily needing to respond to this message. And so the only cells that are going to respond are those cells that express these receptors, right? The only cells that are going to bind the ligand to the receptor. Okay, so a target cell is a cell that expresses the appropriate receptor. Now, of course, that receptor has to have the right shape. It has to have the right structure. And so if there is a mutation in that receptor or in the gene for that receptor, even though the speaking cell is sending the message, it might still not be able to talk to the target cell. Right? Again, protein structure, the genes that they come from, wildly important for the function of the cell, the function of the body. The last type of signaling is essentially talking to yourself. So auto, right, means self. So an automatic car automatically, like itself, changes gears, right? So autocrine is essentially talking to itself. So here we see the ligands, and they are released out into the local environment, and they come back around and they bind directly to the receptor on itself. These are the 4 types of signaling by ligands based on how far away the cells are. And so in our example of a damaged tissue, here we see the damaged skin. So the epithelial tissue is damaged, as well as the connective tissue is damaged. The surrounding cells, as well as some of the cells within the blood, release growth factors. They release ligands that essentially say, it's time to grow. It's time to make more cells faster than we usually are, than we are doing just to kind of maintain the status quo, maintain the skin as we know it. We need to make more because now there's this huge gap and the body is vulnerable. We need to fill in that gap really soon. And so what happens is that a growth factor binds to its receptor on one of these surrounding cells here. And that actually induces a chain reaction of a signaling pathway called a phosphorylation cascade inside the cell. And what this cascade does is it essentially amplifies the response so that we get a really big response really fast. Okay. The pre-programmed response here would be to turn on genes, to activate genes that are involved in the process of mitosis. Okay, so starting to prepare for the process of cell division. Again, we need to make more cells to fill in that gap. And so just as a reminder, mitosis is the process of cell division. In this life cycle of a typical cell, so this might be an epithelial cell within our epidermis, those cells are normally just like kind of living their life, right, doing their thing. But as soon as they receive that growth factor, that ligand signal that it's time to divide, what they're going to do is they are going to begin what's called S phase. They're going to go from growth phase, normal old everyday life, to S phase. And so S phase is the phase in which DNA synthesis occurs. And so what's going on there? Of course, to prepare for mitosis, that is, one cell makes a copy of itself, divides into 2 genetically identical diploid cells. Right? So where there was one skin cell, now there are 2 skin cells. In order to do this, we need to make a copy of our DNA so that both cells have the same amount of DNA and have the same DNA as the original cell had. And so what happens in the process of DNA synthesis is that there is a little enzyme called helicase that breaks the hydrogen bonds between the double helix strands of DNA, and different enzymes called DNA polymerase, right? Very similar to RNA polymerase, but instead of making RNA polymers, these make DNA polymers. DNA polymerase makes a copy of each of those strands, and that copy isn't RNA, right? It's DNA. And so we end up with two identical copies of the DNA. Now the thing about this is that this is happening super fast. Keep in mind that we have about 3.1 billion nucleotide pairs, base pairs of DNA in our genome. And so every time we make a copy of a cell, we need to make a copy of billions of bases, right? So this is a wildly fast process. Like if you think about how quickly your cut heals, right? In a couple days, you have regenerated literally thousands of cells. So this is happening thousands of times. And so this process is kind of prone to making mistakes, right? So instead of, DNA polymerase seeing, this is a cytosine, maybe instead of copying with a guanine, maybe it's a thymine or an adenine instead. And so, as we know, any kind of mutations, any kind of errors can produce or can result in a problem with the protein and therefore a loss of function of the protein. And so these types of mistakes, these copy errors are really, really bad for us, right? They can produce some pretty bad side effects. And so we also, as part of this process, have some proofreaders, right? So other enzymes that come along and make sure that all A's go with T's, all C's go with G's. They see the wrong nucleotide, they quickly replace it. Okay? So that is kind of built in to this process of DNA synthesis. Of course, if there are a lot of mutations, those proofreading enzymes just can't keep up, and some enzyme or some mutations will be passed on to the daughter cells. Okay. Also, as part of this phase, there's a checkpoint. Actually, there's a couple other checkpoints in this process. A checkpoint that essentially looks at the DNA and says, yep, that's good enough. We can proceed with cell division. Okay. All of those mechanisms, the copying, the editing, the checkpoints, all involve proteins, which of course involve genes. Hold that thought. After the DNA is copied or synthesized, the cell continues on to grow some more organelles, right, to duplicate everything within the cell so that we can produce two cells from one. Once we made a copy of everything in the cell, we begin the process of mitosis. For this class, I am not going to ask you to memorize all of the different phases of mitosis for the lecture. Okay. The process of mitosis, just as a summary, essentially involves kind of packing up the DNA, which is usually in its chromatin form, and of course it's just copied, so it's everywhere. Packing them up into little chromatids, little pairs of our chromosomes, lining them up in the middle of the cell along kind of the equator of the cell, using cytoskeleton fibers to pull those copies to opposite sides of the cell, to opposite poles of the cell, and then using actin filaments to pinch off the cell right in the middle to finally produce two independent cells. The actual division part, now we have two independent cells, that's called cytokinesis. And so in a tissue like this, like epithelial tissue, like connective tissue, so these tissues within the skin, they are able to what's called regenerate. Okay. This gap, this damage is essentially filled in via mitosis and a little bit of restructuring of the damaged tissue. And more or less, it looks about the same as it did before the injury. Of course, as I said before, it might be have a little bit of a different color. It might have a scar afterwards, but generally, it's still the skin. And so this process is called regeneration, replacing damaged tissue with the same type of tissue. And this is something that's only seen in epithelial tissues, connective tissues, and smooth muscle tissue. So the tissue that kind of wraps around a lot of the tubes in our body. So our digestive tract, for example. On the other hand, not every tissue can regenerate, unfortunately. For example, this is our cardiac muscles. These are cardiomyocytes. This is what allows our heart to contract and ultimately pump blood out of the heart and throughout our body. If there is damage to the heart, right, so here's an injured area, those damaged muscle cells are not replaced. Okay? They're not replaced with muscle tissue, that is. And of course, muscle tissue is contractile. It can squeeze and therefore pump. Instead, it's replaced with fibrous connective tissue. So it is replaced with a scar instead of contractile muscle tissue. And so what this means is that this part of the tissue is no longer as functional. It doesn't have the same abilities as the former muscle tissue. Scar tissue doesn't contract. And so now the heart is just that much weaker because of the scar. And so we see this process called fibrosis in cardiac muscle tissue, skeletal muscle tissue, like if your skeletal muscle cells die, that's it, you can't grow them back, and our neurons. Right? So if your neurons actually die, you can't make new neurons. So in the healing of a wound, in regeneration, the mitosis is going to continue at this accelerated pace for as long as it takes to fill in that gap. And as soon as those cells start to like butt up against each other from the opposite side of the wound, they're going to go back to the normal pace, right? We're not going to keep undergoing this really, really fast mitosis and like your little cut just gets higher and higher and higher and higher. That's not what happens, right? So this rapid cell division only continues as long as the cells are trying to fill in the gap. But as soon as they touch each other, which is something called contact inhibition, they slow down again. And so what this means is that we have pretty much an ideal rate of mitosis, an ideal rate of making more cells, kind of to replace old cells, replace damaged cells. In our normal skin, normal healthy skin, mitosis for the epidermis is always happening at this deepest layer here. And we're going to go into that today. So it's always happening down here, making new cells. One of the daughter cells stays behind, one of the daughter cells gets pushed up. And so we keep on making those daughter cells and pushing them up and up and up and up and up, migrating superficially. At a certain point, those cells are programmed to die, right? So our skin cells are not immortal, right? They live for a couple weeks and then that's it. This process of programmed cell death is called apoptosis. And what this does, right, it might sound bad, oh gosh, our cells are programmed to die. But what this does is it allows us to continually replace those cells. We make more to replace the ones that just died. And so the surface of our skin, the epidermis, is always only a few weeks old. So it's brand new. These cells are really they're raring to go. They are good at doing what they do because they're young and vibrant, right? So it's a good thing that we can continually replace these cells. But the thing is, the rate of mitosis, making new cells, needs to match the rate of apoptosis, right? We need to make as many cells as are dying off. If we are making too many cells, it's bad. If we're making not enough cells, it's bad. Always a Goldilocks situation. We're shooting for just the right balance between mitosis and apoptosis. And so what happens if that's not the case? All right, what happens if there isn't a balance between mitosis and apoptosis? Well, first of all, how would this happen, right? How would this balance get thrown off? Well, of course, mutations, right? If there are mutations in the genes that code for proteins involved in those mitosis checkpoints, right? Making sure that this cell is ready to go, right? It has the accurate DNA sequence, right? Everything is all well and good. And those proofreading mechanisms as well. So DNA polymerase makes mistakes all the time, but we don't have mutations all around our body because of these little proofreaders that come along afterwards and make sure that the DNA sequence is actually accurate. All right, so if those proteins are messed up because there's mutation in the gene, all of a sudden we are passing on mutations. We are letting cells go and make more of themselves that are filled with mutations. Also, if cells aren't responsive to apoptosis signals, right? So if those cells that are programmed to die at a certain point just don't die, that's a problem too. Right? So in this case, in either one of these cases, cells can continue to divide and they can actually pass on and accumulate more and more mutations. So what we can see in this image is a layer of epithelial cells. They are all of approximately the same shape and size. They're not all crowded together, a nice, even, nice layer of them. But one of these cells acquires a genetic mutation, right? And this mutation can be involved in anything, but especially if it's involved in those mitosis checkpoints or response to apoptosis signals. And so this cell can continue to divide, right? Even though it's touching these other cells, right? It shouldn't be making more of itself. It is like this tissue is intact, but it's unresponsive to the normal cell signals. And so it starts to divide without control. Okay. And so it is going to pass on these mutations, right? Like when DNA synthesis happens in these cells, it's going to copy the mutations. And maybe if those mutations are in the proofreading mechanisms, like it's going to pass on even more mutations. Okay. And so we start to see that this one cell that was that had a mutation makes more and more of itself, and that it doesn't stop when it contacts other cells, and so it makes more and more and more. We see that this little mass is starting to protrude out from the rest of the tissue. This process continues, right, from just overgrowth, hyper, too much growth, to like it's dysfunctional, right, bad growth. When these cells, which of course are dividing out of control, start to accumulate even more mutations. And so these cells really don't look anything like these other cells. They can't be doing the function that is required of this tissue. And so this mass of cells starts to kind of act out, right? It is not doing what it's supposed to be doing. And so this can continue, of course. This tumor, this mass can develop into cancer, of course. So this is a tumor with lots and lots of different mutations. These cells are dividing rapidly. They are spreading on their mutations, their lack of function that is appropriate for this particular organ. And so this tumor is hungry, right? It needs a lot more energy than the surrounding tissue because it's just growing so fast. And so it can actually recruit blood vessels to grow towards it, to have its own blood supply, or it can even grow towards the blood supply. And so this feeds the tumor, but it also means that some of these mutated cells can break off and be transported in the blood and can invade other parts of the body. So that would be metastatic cancer. Okay, so at this point, I hope that you guys can appreciate that cells work together as tissues. Cells hear messages from other cells within the tissue, and they can work together. They can change what they're doing in order to keep the tissue intact. As far as tissues go, we can classify these tissues, these groups of cells working together, in one of 4 different classes, just like the biomolecules, one of 4 different classes. These are the epithelial, connective, muscle, and nervous tissues. Epithelial tissues cover things, they line things. Connective tissues do exactly that. They connect things, they form structure, they form glue, they form all sorts of different things. Our blood and our bones are also connective tissue. Muscle tissue is contractile, so it changes shape actively, and nervous tissue sends lightning fast electrical signals for our nervous system. Okay, so we are going to be talking about all 4 classes of these tissues with a heavy emphasis in Unit 1 on epithelial and connective tissues. In Unit 2, there'll be a heavy emphasis on muscle and nervous tissue. But keep in mind that in our organs and our organ systems, there are usually representatives of all 4 types of these tissues. And so we are going to explore how tissues work and how tissues work together as part of the integumentary system. Right, so here will be our example how we are going to navigate through learning about the tissues. The integumentary system, our skin, is essentially the protector of the body. It forms a barrier against the outside world. It keeps out pathogens. It keeps out, damage from wind, prevents dehydration, so it keeps moisture in and the bad stuff out. Protects us from chemicals. It insulates the body, right? So it can help us to regulate body temperature. The integumentary system can even help us to regulate calcium in the body through kind of an indirect pathway, but the integumentary system can synthesize vitamin D, and vitamin D is critically important in absorbing calcium into the blood from our intestines. And so we are going to be looking at these different classes of tissues as part of the integumentary system and part of the rest of the body. We are going to be doing so via images from the textbook and other sources, but also you should be prepared to learn to recognize these tissues under the microscope for the lab. And so we're hopefully going to be training our eyes to see the differences in tissues. And of course, when you first look at this histology, this tissue slide, it just looks like a bunch of pink and purple, right? But hopefully we can start to see over time that there is, kind of a smoothness to this tissue up here. This is epithelial tissue. This gets a little bit patchier and there are fewer cells, right? This red down here, that's going to be some connective tissue. And this down here, we can see lots of little white circles. That is loose adipose connective tissue. So these are the fat cells. So hopefully we can start to see at least the texture differences, maybe some color differences at this point. And in the lab, we'll dive in a little bit more. So before we jump into talking about the structures and the tissues of the skin, let me first clarify what I mean when I say what is a structure and what is the tissue. So a structure or a layer or a bone or a bump on a bone, right? Those are the things themselves, right? So that is the shape, the structure of the tissue. So it's an item, right? Finger, right? A structure. But what is it made of, right? Like what is a finger made of? It is made of different tissues, right? It's made of bone, it's made of muscle, it's made of epithelial tissue, connective tissue, nervous tissue. It's made of all these different tissues, right? So let's think about that with some analogies. When we wear clothing, we are wearing structures, right? So we can wear a t-shirt, we can wear a golf shirt, we can wear a sweater, right? So those are the things, and there are distinctive differences between those things, right? Sweater, t-shirt, golf shirt, because of the way they're shaped. And so in the same way, we have many structures in the body that have different names because they have very distinctive features to them. So we give them different names. And these features, these structures are made of some kind of material. So the material is cotton, right? The t-shirt is made of cotton. We wouldn't say, oh, like I'm wearing a cotton today. Nope, we would say I'm wearing a t-shirt today, but we can look at the tag and say, yep, that t-shirt is made of cotton, right? And that is the tissue. Of course, if we look under the microscope, we can see that that cotton has individual fibers, right? Fibers from the cotton plant, right? So there's going to be a lot of like cellulose and stuff in that t-shirt. But the tissue itself, the cotton, right, would be the tissue. And of course, these different structures, t-shirt, golf shirt, sweater, have different functions, right? One's better as an undershirt, one is better as an overshirt to like keep you warm. And the different tissues, cotton versus wool, give rise to the function of those different structures, right? You wouldn't put the t-shirt on over the wool sweater, right? Because of the tissue it's made of. Another example, here is a wall. On the inside, like what we would paint, put pictures on, whatever, that is sheetrock, nice and smooth, takes up paint pretty well. We have kind of like scaffolding in the middle made of wood. a stud wall, and we have brick on the outside, like really durable to the elements outside. And so each of these parts of the wall, we call them something different, right? Sheetrock, stud wall, brick wall, and they're all made of something different. And the tissue, of course, gives rise to the function, right? The material, what's made of, gives rise to the function of the structure. We wouldn't put sheetrock on the outside of a building because as soon as it rained, it would just fall apart, right? So the structure and its tissue matches the function. All right, so let's take a look at the integumentary system. The integumentary system is an organ system. That is, it has multiple organs that are made of tissues. The integumentary system can be divided into two distinct regions of skin, and then under that, there is a third region. Okay, so the skin itself, or the cutis, is made of or consists of the epidermis, the most superficial part. And so that's the structure, but then it is made of, right? T-shirt is made of cotton. Here, the epidermis is made of epithelial tissue. We see epi twice here. Epi means on top of or upon. And so epi, this is upon, it's on top of the dermis. The dermis, right, is just deep to the epidermis. And so this is made of connective tissue. So both of these things together are what we consider to be our skin, right? Deep to the skin is another layer, another structure. This is called the hypodermis. So hypo means less than or underneath. So hypodermis is a subcutaneous layer, right? If the cutis is the epidermis and the dermis, a sub under cutaneous and under skin layer is deep to the skin. For the most part, the hypodermis is made of adipose tissue, loose adipose connective tissue, and this is really important in shock absorption. Right, so essentially it provides like a little bit of padding for the underlying tissues, the muscle, the bone. Of course, when this layer gets really thin, so you lose a lot of that subcutaneous fat, you know, your bones kind of protrude a little bit more, and it's really easy to like bump them on things and hurt yourself, right? So this extra little padding is actually protective. Insulation, so this fat layer is important in insulating the body temperature. And it anchors the skin to underlying structure. So make sure that the skin is actually attached to your muscles underneath. Another term for the hypodermis is called superficial fascia. Okay. And so as we're going to see through this semester and next semester, there's just connective tissue that forms this network that's actually interconnected throughout your entire body, this fascia. And so there's a little bit of like movement of fluids and movement of ligands and stuff around in this fascia. And the fascia essentially like connects all of your body all together. And so the most superficial of that connective tissue, that most superficial of fascia is the hypodermis, right? Keeping your skin attached to the rest of your body. So let's zoom in on the epidermis. That's what the rest of this lesson is going to be about today, the epidermis and what it's made of. The epithelial tissue can also be called epithelium. It can be found covering things, lining things, right? So covering the surface, of course, the epidermis is the most superficial part of our body. It covers our body itself. It also lines structures. And so here we see the stomach. And if we take a section through the stomach wall, we see that there are epithelial cells, right? Lots of cells squished together here. And so this forms the lining that would kind of interact with your food, interact with the stomach acid of this tube. Okay, so it lines structures. And so on a test, either a practical or a lecture exam, if I ask, what is the tissue that is lining the kidney tubule? What is the type of tissue that is lining the alveoli? Right? You automatically know it's an epithelial tissue. Another thing that epithelial tissues are good at is secreting, right? So they form glands in many cases. One of the functions, epithelial tissues are great at kind of a protective function. They form a great barrier. So these cells, as we're going to see, are kind of sewn together by different cell junctions. So the cells are right up against each other. They are sewn together, and in order for something to get past this barrier, they have to go through the cells. And as we know, these cells are surrounded by phospholipid bilayer, which repels pretty much everything. All right, so it repels hydrophilic stuff, it repels hydrophobic stuff, and so it is super selective. Okay. So as this barrier, this selective cellular barrier, this protects our body in an immune way as well. It keeps stuff from your stomach from leaking out into your abdominal cavity. It keeps stuff from outside your body from getting inside your body, inside your skin. And so there's a lot of immune defense to this barrier as well. Of course, epithelial tissue can secrete stuff in many cases, etc. Okay, what are the features of all epithelial tissues? All right, so these are the functions, is where we find it, but what epithelial tissues look like? Epithelial tissues, unlike some of the others, are mostly cells. So it's less protein stuff, less fluid stuff outside of the cells, and more just cells. Right? When we look at this, it just looks like a solid block of cells. These cells, because they form a barrier, they form a covering or a lining, they have what's called polarity. So here is a bit of epithelial tissue. This is the outside world, or the lining of a tube or something. This is like the rest of that organ, the rest of the body. And so some of the cells are on the top, they face the top, and some face the deeper side. And so we can say that these cells have essentially a top and a bottom. The apical surface points into the lumen, into the opening of the organ, or outside the body. And the basal surface points deeper into the wall of the tissue, or sorry, the wall of the organ, or into the body. And so the basal surface of the deepest layer of epithelial cells is always attached to this little bit of connective tissue called either the basement membrane or the basal lamina. The cells themselves are connected to each other, and they're connected to other tissues. What we can see in this image are 3 adjacent epithelial cells forming that nice barrier. Epithelial cells can be and are often joined together by what are called tight junctions. So these are the ones that literally sew the cells together and make it so nothing can pass in between the cells. If something wants to cross this barrier, it has to go through the cells. And in order to go through the cells, the cell has to let that thing through. Desmosomes, these are plaques, right? Like protein plaques in one cell and the adjacent cell, and they are sewn together in the middle with like super strong proteins. And so what these function in is keeping that membrane intact, keeping it together. Of course, as we move our body, as we move our organs around, those epithelial tissues must flex and extend a little bit. And so we don't want them to tear apart, right? So we sew them together with desmosomes, which are super strong. Gap junctions, these are more like little tunnels. So this essentially connects the cytoplasm of this epithelial cell to the cytoplasm of this epithelial cell, allowing substances to pass right through into the neighboring cell without having to go outside the cell and then come back inside the cell. This is like a fast track, a tunnel from one to the next. And so this also allows epithelial cells to work together as a tissue. And so these 3 cell junctions allow the cells to be bound together to other epithelial cells. A hemidesmosome essentially sews the epithelial cells to the underlying basal lamina. So the deepest cell has one of these little protein plaques like we see over here in the desmosome, and the little protein fibers not sticking out into another little plaque in another cell, but instead forming a little anchor point into the connective tissue of the basal lamina or basement membrane. All right, some more features of the epithelial tissues. Epithelial tissues are avascular. In other words, they don't have any blood vessels. And so that has some implications for the structure and the function of epithelial tissues. Blood, as we know, is required to deliver oxygen and nutrients for the function of metabolically active cells. If there is no blood that is passing into the epithelial tissue, it means that those cells can only get the good stuff, the oxygen, the nutrients, when those things diffuse from the blood nearby through one cell and another cell and another cell and another cell. And so this process takes forever. So epithelial tissues, for the most part, remain pretty thin because anything that's too thick can't get what it needs from the blood because the blood is just so far away from it. The exception is the skin, right? And the epidermis is a little bit thicker than most other epithelial tissues. And the way that it can get away with that is that most of the epidermis, a good portion of the epidermis, is actually dead cells, right? Remember that our cells are programmed to die at a certain point, at a certain age within our skin. And so all of the cells that are farthest away from the blood, they're dead. They don't need anything from the blood. In general, epithelial tissues are really thin because they're avascular. They have no blood vessels. The epithelial tissues are named according to the cells that are in them. They are named according to the number of cell layers and the shape of those cells. The simple epithelial tissues have a single layer of cells that attaches to the basement membrane. Stratified has many layers of cells, or at least 2 layers of cells. And in some cases, we have pseudo-stratified, where technically all of the cells are attached to the basement membrane, but they have all sorts of different shapes and sizes, and so it kind of looks like they're stacked on top of each other. The shape. Squamous cells are flat, they're squished, they are scale-like. Cuboidal cells are cube-shaped, they're about as tall as they are wide. And columnar cells are much taller than they are wide. So they're little columns. We also have transitional cells, which sometimes look a little bit more cuboidal, and sometimes they're stretched out to look a little bit more squamous. So in this lesson, we are focusing on the skin, specifically the epidermis. The epidermis, what is it made of? It is made of stratified squamous epithelial tissue. Stratified because there are many layers. Squamous because the cells are a little bit flat, right? Especially when we get way up here to the top. Again, why does this tissue make sense, right? Why does brick make sense on the outer wall, but sheetrock makes sense on the inner part of the wall? Well, this tissue makes sense for this structure because the many layers of cells form a really thick protective barrier. Right? If you are a bacteria, it's going to be really, really hard to wiggle yourself through this entire barrier. Right? The other thing is we are constantly like brushing up against things, right? Like, clothes are rubbing on our skin, whatever. And so the fact that we have so many layers of these cells, and in fact, so many dead layers of these cells, means that we are constantly like sloughing off some of those cells. I'll talk more about that later. And if we do lose some cells on the outside because we scraped them off, there's still plenty of cells left to that layer. Squamous. Why does squamous make sense? Not cuboidal, not not columnar. Squamous cells are essentially just a phospholipid bilayer with very, very minimal stuff in the cytoplasm, very minimal organelles. And so these cells aren't really doing very much. They are just little sacs that form a shell, but they're kind of cheaply made, right? Like we don't really invest a lot into making a flattened squamous cell. So again, if we just slough some of those off, if we lose some of them, no big deal. We didn't actually invest that much in them. The other word here is keratinized. Right? So keratinized means that this tissue has keratin in it. Not all stratified squamous epithelial tissue has keratin in it. Only the outermost ones, the most superficial stratified squamous epithelial tissues. And so keratin is a protein. We heard of it when we talked about the cytoskeleton. This is a fibrous protein that essentially makes those cells really tough. And so if you are a cell on the very surface of the body, you are exposed to all sorts of things. And so you need to be really strong against it. Other epithelial tissues that are within our body, they don't need to be quite as strong. They're protected by the rest of our body. And so they would be not keratinized. Again, organs like, or essentially any organ that's exposed to the outside world, like our oral cavity, right? We are breathing in through our oral cavity. We are taking in food, which is a lot of times like very scratchy. And so it scratches the lining of our oral cavity, of our mouth, right? It scratches our throat, our esophagus on the way down. So all of this tissue, the lining, is also stratified squamous epithelial tissue, but the cells are not keratinized because these cells are always moistened. They're always covered with saliva or with mucus, so they're always moistened, and so they do not need keratin. All right, so the epidermis is made of stratified squamous epithelial tissue, which happens to be keratinized. The epidermis itself is broken into 4 or 5, depending on where we're looking, sublayers. All right, so these would be structures. On a practical or on a lecture exam, I might tell you sublayers or minor layers. Like, what are the different layers within the epidermis? And so in this image, and in a lot of images that we're going to look at for the rest of the PowerPoint, we see a drawing of what a section through the skin is going to look like. And here is what the histology looks like. So this is literally this, but drawn out so we can see things a little bit more clearly. Okay, so what are the layers? Going from superficial to deep. So if you were giving an injection, right, what layers would the needle pass through? The first layer would be the stratum corneum. So stratum means layer, stratum corneum. If you were giving an injection through the palm, which you surely wouldn't be, or the sole of the foot, there'd be an extra layer of the epidermis. This is called the stratum lucidum. I'll talk more about that shortly. Next is the stratum granulosum, stratum spinosum, and stratum basal. And so, yep, lots and lots of different terms, but I have a little mnemonic for you guys. That mnemonic is, come, let's get sunburned, okay? Corneum, lucidum, granulosum, spinosum, and basal, from superficial to deep. And again, unless you are talking about skin in the palms of the hands and the soles of the feet, it would become get sunburned, right? Lucidum is only present in those 2 places. I'll remind you of that. All right, so let's actually start from the deepest and work our way up, because this is where cells are born, right? The cells of the epidermis are born. So the stratum basal is a single row of cells that happen to be actively dividing almost all the time. They are stem cells that can differentiate, right? They can grow up to become regular old epidermal cells or a couple other things, but these are little stem cells. Cellular programming causes these cells to divide at a particular rate, which essentially matches the rate of apoptosis, like I talked about earlier. So this is kind of a section through the epidermis. This is one of those stem cells within the stratum basal. It undergoes the process of mitosis, producing 2 daughter cells. One of those daughter cells, A, is going to remain behind and it's going to stay in the stratum basal, and it's going to keep on dividing, keep on making more and more cells. But the second cell, the B cell here, we can actually follow it, right? So this one is going to become a normal old epidermal cell called a keratinocyte. So cyte means cell, and keratin, this is a cell that's chock full of keratin. Makes sense for a keratinized epithelial cell. Okay, so as cell A keeps on dividing, it makes more and more and more cells. And these brand new cells are going to push this first B cell more and more and more superficially. And so in this way, our epidermis is made in stratum basal, and it grows up and out. It grows more superficially. Now, over the life of a keratinocyte, it is going to go from this nice big rounded cell, and it's going to flatten out kind of until it dies, right? During this process, though, as it is migrating superficially, as it is flattening out to become a true squamous cell, it's making lots and lots of keratin, right? It is a keratinocyte after all. And again, it is programmed to die at a certain point in its migration. There are other cells in the stratum basal. The first cell I want to point out are the melanocytes. So here is one of those stem cells, and a stem cell, and a stem cell. All of these are the stem cells that are actively dividing. But kind of embedded within that stratum basal are these big, gangly, multi-limbed melanocytes. And as the name suggests, it is a site of the cell that makes melanin. Melanin is a pigment, and there's a couple different types of this pigment. Usually it is brown or yellow brown or black, so different shades of this melanin determined genetically and also determined by how much you've been out in the sun. Okay, like at least how much is made, and essentially these melanocytes are actively synthesizing melanin, this pigment, packing it into little vesicles called melanosomes. And right here's a melanocyte making lots and lots of melanin, packing them into little vesicles called melanosomes, and releasing those melanosomes, which are then taken in by the keratinocytes. All right, so we can look up here as well. This melanocyte is releasing melanosomes, right? So dumping off lots of melanin in the surrounding keratinocytes. Okay, and so what's the point of melanin? Like, why is this happening? Melanin essentially acts like a little umbrella. All right, so in the accepting keratinocyte, here we see the melanin is forming a little umbrella on the apical side of the cell. And what this little pigmented, this colored umbrella does is it essentially absorbs UV rays, protecting the nucleus, which of course has our DNA, from the sun. The UV rays, as I'm sure you're aware, UV rays damage the DNA, they mutate the DNA, and we'll talk more about that in a future lesson, but UV rays mutate the DNA. We know at this point, hopefully, that mutations in the DNA lead to some really bad consequences, and so it's really important to protect our DNA from the UV rays. And so melanocytes are actually sensitive to the UV rays that we expose ourselves to. So the more you're out in the sun, right? Someday you'll go back out and you will sit on the beach again, and you'll be exposed to lots and lots of UV rays. That stimulates your melanocytes to make more melanin, right? Oh, like these cells know, yep, we're exposed to more of these damaging rays. We need to kind of thicken the umbrella to make sure that our DNA is protected. All right, so this would appear as a suntan. Note that melanin doesn't protect the DNA entirely, right? So even if your body produces more melanin, even if your skin tone is a little bit darker because you have more melanin, it isn't a fail-safe, right? And so it doesn't protect your DNA entirely. Even if you have a darker skin tone, it's still a good idea to wear sunscreen to protect your skin and thus protect your DNA, because melanin isn't perfect. So, of course, melanin, these pigments, are very influential over our skin tone, but they're not the only ones. The only factors determining our skin tone. We also have other molecules called carotene, and yes, these carotenes are found in carrots, yes. So the amount of carotene you have in your skin also slightly influences your skin tone. Hemoglobin as well, particularly in individuals that do not synthesize a lot of melanin. So, like a fairer skin tone, it is a lot easier to see blood vessels just deep to the epidermis. And so, the pigment in our blood and our blood vessels is hemoglobin. It's reddish, and so this red can influence the skin tone as well, right? Like when you're blushing, you are seeing more hemoglobin. The other type of cells that are found in, found in the stratum basal are part of the nervous system. So this is actually nervous tissue. Here we see the epidermis with all of those epithelial cells. This purple layer down here is the stratum basal, and embedded within the stratum basal are these little tactile cells. Formerly, they were called Merkel cells, named after the guy that discovered them, described them, whatever. For the most part, in A&P, we are kind of trying to like steer away from naming things after like old anatomists, old scientists, but you still will hear either one. So I will give you both terms. So tactile or Merkel cells are these little cells embedded within the stratum basal that are sensing really fine touch, right? So like really faint stimuli on the surface of your skin. And we can see that these Merkel cells, when they are slightly distorted by your, by slight light touch, they send a signal along this nerve, along this neuron, up towards the brain so that we know that we are sensing some kind of light touch. Strata, granulosum and spinosum. Strata is plural, stratum is singular. So we're going to look at two strata here. Building upon our diagram here, the deepest single layer is called the stratum basal. Moving more superficially, let's see the stratum spinosum. Stratum spinosum is usually the thickest layer. So all of these thick, somewhat round cells, like they're a little bit rounder than the diagrams of squamous cells look, but we still call them squamous. It's called spinosum because they're spiny, they're spiky. Essentially, these cells have a lot of filaments attached to the desmosomes that are holding them together, maintaining that nice barrier. So when you look under the microscope, it might seem like these cells are a little bit spiky. These are the very metabolically active cells. They are the closest really to the blood supply. We usually have blood vessels that are like right next to the epidermis. And so these guys can do lots and lots of stuff. Namely, they're making a lot of keratin, right? They are keratinocytes and they're making lots and lots of keratin while they are still close enough to the blood to get their resources. Also, these cells synthesize vitamin D. Within the stratum spinosum, we also have some immune cells that are constantly like weaving in and out, crawling over the spinosum cells, the keratinocytes. And essentially, they're looking for anything that doesn't look quite right. If there is an infection, right? If a bacteria has wiggled its way all the way through these cells up here, we don't want it to get into the blood. Once it's in the blood, it can affect the body. So these dendritic cells are constantly looking for any kind of invader that have made it this far. If there is something, it's going to engulf, it's going to phagocytize, engulf that invader, and it's going to show it to the rest of the immune system. Next up is the stratum granulosum. It's called granulosum because when we stain this tissue, and so then it'll look pink or a little purple, these granules, which contain keratin, and they contain like a little waterproofing molecule that is lipid in nature. These granules take up a really dark stain. The stratum granulosum is the last layer in which the cells are actually alive. They are programmed to die at this point. and it'll be the darkest layer. A little bit hard to see here, but the darkest line, right, pretty thin line, is the stratum granulosum. Stratum lucidum, called lucidum because lucid means clear. So if you are lucid, you are thinking clearly. This is the one that's only present in a couple places. It's a pretty thin layer and it's pretty clear, right? So this is a clear layer. It doesn't actually take up stain like the rest of the layers of the epidermis do. And the stratum lucidum is what gives some skin its kind of gummy texture. So on your palm, like it's a little bit gummy, right? A little gummy. And so that's because of the stratum lucidum. This tissue is, or sorry, this layer is only found in the thick skin. And so I want to emphasize that there is thick skin and there is thin skin. The thick skin is found in your palms and the soles of your feet, right? So where, that gets a lot of wear and tear. The thin skin is pretty much everywhere else, right? It's on your face, it's on your back, everywhere else, not your palms and your soles. The thick skin also has some other features. It is also characterized by having a really thick stratum corneum, so the most superficial layer. In this bit of thick skin, we have the stratum basal, the stratum spinosum, granulosum is that nice dark line, and then a really thick corneum way up here. In this thin skin, again, basal is a single layer, spinosum is relatively thick layer, the dark line is granulosum, and then this is the stratum corneum, so much thinner. Thick skin is also characterized by having no hair. Okay, so there are at least fine hairs on every bit of skin in your body, except for the palms of your hands and the soles of your feet. Again, palmar and plantar, palms and soles, that's it. So that is the stratum lucidum. The stratum corneum is called corneum because it's cornified. It's kind of like scaly and flaky. This layer is only dead cells, like purely dead cells in the stratum corneum. These cells are super flat, so like true blue squamous cells at this point. Their nucleus at this point has died. They are more or less just sacs full of keratin. And we want this because this is a great barrier. If some of these dead cells flake off, doesn't matter, they're already dead, it's all good. Again, they are functional, even though they're dead, the keratin is still very helpful. It's still a very good protective barrier. It protects our cells from the environment, from being scraped up, from invasion by pathogens, and it keeps the water in. It is this layer of cells that is constantly being sloughed off at the rate of about 50,000 cells per minute. So we do lose lots and lots of cells from the stratum corneum, but fear not, we are constantly making replacement cells down here in the stratum basal. And so I give you guys this image just as a summary of all the stuff that I just talked about. Here we have the thick skin and the palmar and the plantar only. Here we have the thin skin, which is literally everywhere else. Some key differences are the stratum lucidum in the thick skin, but not the thin skin. There is a thicker stratum corneum. We have these dendritic or Langerhans cells in both. Here we have the melanocytes and the multiple strata. And so that's just for you to summarize all the stuff we just talked about. So at this point, we've talked about the epidermis. It's made of stratified squamous epithelial tissue that happens to be keratinized. But this is not by far the only epithelial tissues in the body. Now I would like to walk you through the other epithelial tissues. For the lecture, you should be familiar with what tissues are found in what organs, right? So I can ask you what is the, what type of tissue makes up the serous membranes, lines the alveoli of the lungs, like whatever. So you should know where it's found and what tissue it is. You should also be familiar with why that makes sense, right? Like why does simple squamous epithelial tissue make sense in this place? Okay, so the function, the structure, where it's found. Okay, for the lab, the questions are more of just what is this tissue under the microscope. Okay. So simple squamous epithelial tissue, a single layer of really flattened cells. Here is a drawing, and here is the histology, right, to help you kind of parse out which is like what you're seeing in the histology. Simple squamous epithelial tissue is the thinnest possible barrier that we can make, and therefore it has rapid diffusion, right? We can let things go across this membrane really, really quickly. And so this makes sense in the serous membranes. Here we're looking at the pleura on top of the lungs. Okay. Up here we can see other serous membranes, so very, very flat, skinny cells are making up this thin membrane. Also, the alveoli of the lungs. So every time you take a breath in, that air is going all the way down into these little spheres called alveoli at the base of your respiratory tree. And the goal is to get oxygen across this barrier and carbon dioxide across this barrier in opposite directions, lightning fast. Like as soon as we take a breath in and out, all of that diffusion has happened. And so if we had a really thick barrier, it would take forever for gases to actually diffuse. And so a really thin membrane makes sense for places where we really, really want rapid diffusion. Simple cuboidal epithelial tissues. Cuboidal cells are thicker, they have more cytoplasm, so there's more space for organelles and all the things that organelles do. Cuboidal cells are great at filtering. They're also great at secreting stuff. And so in our kidney tubules, right, in the tubules that are filtering the blood and kind of pulling out the stuff that we don't want, dumping it into the urine, pulling stuff out of the urine and bringing it back into the body, back into the blood. And so there's a lot of like back and forth across these cells, as well as secretion of things like bicarbonate. So if we look at the histology, we see that there's this open space, and we see that there are little cells all the way around it. These are roughly cube-shaped cells, and here the same thing just drawn out for you. Our kidney tubules are lined with simple cuboidal epithelial tissue because they are filtering, they are secreting things really quickly. Also, you will see in the histological slides that there are some of these tubules like this surrounded by simple cuboidal epithelial tissue as part of other ducts, right? So like in the tube that takes sweat from your sweat gland all the way up to the surface of your skin, you'll also see some of these cuboidal cells or simple cuboidal cells. Oops, sorry, this should say simple columnar epithelial tissue. My apologies for that. So simple columnar epithelial tissue over here. These cells are taller than they are wide, and so this gives them lots and lots of space for making stuff, essentially. The pancreas is really good at making chemical buffers. It's really good at making digestive enzymes. And so it needs to make lots and lots of those things and secrete them into the digestive tract. Simple columnar epithelial tissue is also found lining our intestines. And these linings are great not only at secreting stuff, but absorbing, right? So absorbing glucose from our meal, absorbing lipids, repackaging them before they get put into circulation. Okay. So these tissues, simple columnar epithelial tissue, is found lining the intestines, lining the pancreatic ducts. Okay. There is one other type of simple epithelial tissue. This is called pseudo-stratified columnar epithelial tissue. Pseudo because it looks stratified. It looks like there are multiple rows of cells kind of stacked on top of each other, but if you look closely, all of these cells are technically attached to that basement membrane. Okay, so this forms a relatively thick barrier, so barrier against like whatever's in the outside world. Oftentimes, this tissue is ciliated. Okay, so we can see this fluffy cilia up here at the top. And so we find this tissue mostly lining our upper respiratory tract, our nasal cavity, our trachea, our windpipe, our bronchi. And so what is happening here is that, of course, we are taking in air that's filled with particulates, that's filled with bacteria and viruses, and we don't want all of that stuff to get way down into our alveoli. And so what we do is with this tissue, we secrete lots of mucus, right? So that's what these little cells are right here. When you look under the microscope, you'll see just like ovals of white. Those are cells that secrete mucus. We'll get into that soon. So we secrete mucus onto the surface of this tissue, the apical side of this tissue. And what that mucus does, I'm going to switch up here. What that mucus does is it traps foreign particles. Now, once those particles are trapped, we don't want them to just like sit in the lungs and, find a nice cozy place to live. We want to get them the heck out of our lungs. And so what the cilia do is they beat and beat and beat, creating a little current and pushing that mucus with the trapped foreign particles up and up and up and out of our lungs. And then it goes down our esophagus and the stomach acid takes care of it. But we get it out of our lungs pretty well, right? Usually. And so this is called the mucus escalator, very appropriately named. All right. Next, the stratified tissues, stratified cuboidal and the transitional epithelial tissues. The stratified cuboidal, you usually only see like one or like 2 or 3 layers of cuboidal cells around this little lumen in the middle. So this is where the sweat would be made, or milk if it's a mammary gland. And surrounding 2 or 3 layers. If one layer of cuboidal cells is really cool at like filtration and secretion, you can imagine that multiple layers of these cuboidal cells are even better at secreting. Okay. We also have the transitional epithelial tissue. This is the one epithelial tissue that is not 4 words long. It's not stratified cuboidal epithelial tissue, 4 words. It's just 3. Transitional epithelial tissue. So this tissue lines much of the urinary system. So these structures that sometimes are full of urine and sometimes not so much full of urine. And so this tissue needs to stretch. Here we can see kind of a relaxed transitional epithelial tissue, like all the cells are kind of standing up. But we can take this tissue when the bladder is full, for example, and we can stretch it out without tearing the tissue. So those are the types of epithelial tissues, but I want to point out that some epithelial tissues, some cells within the epithelial tissues are secretory, so they secrete lots of stuff. So let's start with the cells themselves. What we're seeing here is part of the intestine. This nice convenient row of nuclei in columnar cells. All right, so this is a simple columnar epithelial tissue, and embedded, kind of wedged in between these normal columnar cells are these kind of bulbous cells called goblet cells. This over here is the lumen, so your food would be here. This over here is the lumen, so your food would be here. So these goblet cells, if we take a look at what's inside, they have a nucleus, they've got lots and lots of rough ER, Golgi apparatus constantly pumping out these vesicles that are chock full of the protein called mucin. And so goblet cells are essentially mucin factories. All of these little things here, these vesicles, are full of mucin that is then released outside of the cell and into the lumen. And so you can imagine that in your intestines, as food is pushed past, right, pushed along this super long tube, over time, the super fluid nature of the food mixed with digestive juices and everything, it's going to get more and more solid, especially as you get closer to the end of the intestines, right? As you get into the colon, water is reabsorbed into the body and the stool, the feces, becomes solid. And so you kind of need a lot of lubrication so that that solid stool doesn't scrape up the lining of your intestines. And so goblet cells become more and more abundant as you get lower into the GI tract. Okay. In addition to specific cells secreting stuff, in this case mucin or mucus, epithelial tissues can form glands. So they're not only for covering things, they are for secreting things. So glands are epithelial in nature, but these specialized secretory cells, right, type of epithelial cells, are going to be the most common. There's two different types of glands, or at least glands can be classified according to where they are releasing their contents. The exocrine glands secrete in an exocrine capacity. That is, they secrete not into the blood. Instead, they secrete into a duct and onto the surface of something or into a lumen of something. So they're not going into the blood, they are going into or onto a surface. So think of your salivary glands. Saliva isn't secreted into the blood. What would be the purpose of that? Instead, the saliva is secreted into the oral cavity. So salivary glands secrete into a duct, which dumps out into our mouth. Sweat glands are exocrine. Right? It wouldn't make sense to secrete sweat into the blood, and so sweat is secreted through a duct and onto the surface of our skin. On the other hand, endocrine glands secrete directly into the blood. Okay, so here, this gland secretes hormones into the blood, which are then carried away to talk to their target cells somewhere else in the body. Glands are not only classified by whether they secrete into a duct or into the blood, but also by how they secrete. And there's a couple different kind of strategies for how these epithelial cells secrete things. Two of those strategies are here. In one type of gland, we see that the cells are pretty much making little vesicles. The Golgi apparatus is making little vesicles, and those vesicles are opening up, releasing merely their contents. Holocrine secretion, the whole cell filled with all of its stuff, everything it needs, is secreted into the duct and up onto the surface or into the lumen. Okay, so merocrine merely secretes the contents, merely secretes. Holocrine, the whole cell is secreted. All right. Thank you guys so much for listening. That is the end of this lesson. I look forward to picking up with our next lesson on connective tissue and the rest of the integumentary system. Thanks.